So the discovery of the reward system in the brain has been one of the most remarkable in the whole of neuroscience. This discovery really was largely helped by studies of drug addiction and the rewarding properties of drugs for abuse, especially in experimental animals such as rats. On the left-hand side, you can see one of the most successful animal models in the whole of neuropsychiatry, that of intravenous self-administration of drugs. So rats will voluntarily self-administer small infusions of solutions into their veins through a catheter containing drugs such as cocaine, amphetamine and heroin. And indeed this self-administration predicts drug abuse in humans. This technique was used by Bartley Herbal in a classic study in the 1980s, where he showed that rats would self-administer amphetamine not only into the vein, but also into the brain. He showed that a particular hotspot in the brain was a structure at the base of the forebrain, in the diagram you can see on the right, a structure called the nucleus accumbens. And this is a particularly interesting discovery because the nucleus accumbens is known to receive projections from nerve cells containing the chemical neurotransmitter dopamine. Moreover, amphetamine and other drugs such as cocaine are known to enhance the levels of dopamine when they're administered. So this led to the hypothesis that dopamine was a reward transmitter, and this was being self-administered by rats directly into the nucleus accumbens. Other drugs for abuse which don't directly affect dopamine may also indirectly affect it, such as nicotine and heroin. So this may have a more general theory in terms of reward systems and drug abuse. Other discoveries have also shown that this system is implicated in natural rewards, in particular in the anticipation of rewards. And moreover, these results have also been shown to transfer rather dramatically to human studies, largely through the use of brain imaging methodology. Now, a classic study by Noel Volkow, who's the current head of the National Institute on Drug Abuse in the US, has used the technique of positron emission tomography to label dopamine receptors in the human brain in the striatum, the same part of the brain as the nucleus accumbens. On the left-hand side, you can see the labeling of dopamine receptors as shown by red and yellow in particular in the human striatum. And these are transverse sections of the human brain. They're cut across, so you're looking below. On the right-hand side, you can see the levels of dopamine receptors in drug abusers, cocaine, methamphetamine, heroin and alcohol, and in every case you can see that they're diminished. So this study was able to show that indeed it's the dopamine system that appears to be dysregulated in chronic drug abuse. But this study raised several interesting questions, particularly related to cause and effect. Are these changes in dopamine receptors in the drug abusers consequences of drug abuse, or is it the case that they may have been occurring before the drugs were taken by these individuals? In other words, they may even have predisposed the drug abusers to taking drugs. This is very difficult to answer from human studies alone because, for example, you can hardly PET scan babies to find out whether they have low dopamine receptors. Another interesting puzzle is posed by the behaviour of some of these drug abusers, which often is very impulsive and risk-taking. This behaviour again could be caused by chronic drug abuse, or it could be pre-existing. and even lead to drug taking. After all, taking drugs is very risky. So which is it? We've been using an animal model which helps to resolve this question of causality. On the left-hand side, you can see rats being screened for their levels of impulsive behaviour. This happens in a test of attention where they have to detect brief visual signals for food. Many of these rats, however, respond prematurely and take risks before the targets are even presented. This proportion of rats, about 10% in the normal population, we label as high impulsives. Intriguingly, when these high impulsives are subsequently allowed to take drugs, for example cocaine, they take very high levels of cocaine compared to the normal or low impulsive animals. And you can see the development of this binge cocaine taking on the right-hand side. These high impulsive rats will even take electric shocks in order to take their drug of choice, in this case cocaine. So this is indeed a very compulsive, addictive like of behaviour. Now, we not only measured the behaviour of these high impulsive animals, but also changes in the brain. And you can see, just as in the humans, these rats, high impulsive rats, have low dopamine receptors in the ventral stratum or the nucleus accumbens. You can see the correlation on the right-hand side. So these rats have changes in dopamine receptors before they even see cocaine, which suggests that the changes are not caused by the cocaine self-administration. And so this reflects back in the human case and asks the question, maybe humans who take drugs also have low dopamine receptors to begin with. Now, these rats also have other changes in the brain, suggesting, for example, that there is a diminished top-down control from their frontal lobes on these subcortical structures, and so their volitional control is impaired. How do these studies translate then to the human situation? In a recent series of studies with Karen Ursher and Ed Bullmore, we've been studying not only drug users, in this case predominantly cocaine abusers and control subjects, but also the siblings of drug users who don't themselves take drugs, in order to get at whether there is a predisposition in these drug users for taking drugs related to impulsivity and changes in the brain. And we've also used a refined measure of impulsivity, the ability to respond rapidly to inhibit a response, thus showing the ability to control your behaviour. This is the so-called stop signal reaction time task shown on the left. Now, if you're slow on this task, it means that you have a degree of impulsivity, and you can see that certainly the drug users are much slower than controls to stop a response in this paradigm. But what is remarkable is that so too do their non-drug using siblings show the same tendency. So this tendency for impulsivity is present in the family, presumably as a result of either genetic or environmental, family-related factors, and possibly both acting together. And we hypothesise that it's this tendency which may lead to taking drugs. Now, what protects the siblings from taking drugs and gives them resilience is an interesting question for further research. This propensity is also associated with changes in the brain. As we see in the brain diagram, when one uses magnetic resonance imaging to measure the white fibres in the frontal lobe, we find that there is a reduction not only in drug users but also in their siblings compared to controls. And this we interpret to mean that there is a loss of volitional control produced by this change in frontal lobe function, which no longer adequately regulates or inhibits the subcortical structures of the striatum, including the nucleus accumbens. Now, this set of phenomena we we label as an endophenotype. So this is a behavioural and neural change which is present in the drug users but also present in their siblings, and may indicate that it's a very basic fundamental contributor to drug abuse and addiction. In this talk, I've tried to illustrate how one can go from animal experimental studies to humans and back again in order to unravel the causes of drug addiction. Thank you very much.